Inside the mitochondria, electron transport chains carry out the following two reactions. In the first reaction, NADH is oxidized to NAD+, and oxygen is reduced to water. In the second reaction, FADH2 is oxidized to FAD+. Oxygen is reduced to water. In both reactions, delta Gs are negative, indicating a large amount of energy is released. This energy is captured by electron transport chain and preserved in the proton gradient. Eventually, this proton gradient is used to drive ATP synthesis. This is oxidative phosphorylation. The question we ask is how these events proceed in the cell. To address this question, in this lecture, we will cover two topics. For the first topic, I will introduce you the relationship between free energy and chemical reactions. For the second topic, we will look into components of electron transport chain and oxidative phosphorylation. Now, let's go to part 1. Given a chemical reaction, A to B, delta G represents change in Gibbs energy, or sometimes called change in free energy. When delta G is negative, such as the reaction shown here, free energy is reduced. We know from organic chemistry, this reaction can occur spontaneously as written. Second, this reaction is exergonic, meaning this reaction will release energy. On the other hand, if delta G is positive, such as the reaction shown here, free energy will be increased. We know from organic chemistry, this reaction cannot proceed in the direction as written. This reaction is endergonic, meaning energy must be added for the reaction to proceed. This reaction can proceed in reverse direction. And we also know from organic chemistry, when delta G equals 0, this reaction is in equilibrium. There are a few important points I want to highlight here. First, giving a reaction A to B, the absolute value of delta G for this reaction equals absolute value of delta G for the reverse reaction B to A. Second, delta G depends on concentration of products and reactants. Delta G can be determined by the following equation. Delta G equals delta G0 plus R times T times natural logarithm of B concentration divided by A concentration. Here, delta G0 is the delta G at standard state when reactants and products are at concentration of 1 molar per liter. R is a gas constant. T is absolute temperature. For a given reaction, delta G0 is a constant. At constant temperature, T is also a constant. Therefore, delta G depends on concentration of reactant and products. This equation is important for understanding change in direction of a chemical reaction. One conclusion we can make from this equation: for any reversible reaction, when concentration of a reactant is greater enough, delta G can be negative eventually, and the reaction can proceed in the direction as written. To elaborate on the first conclusion, here I want to give you an example. Consider a reaction in glycolysis catalyzed by phosphoglucose isomerase. Under a standard condition, when both reactant and product concentration are at 1 molar per liter, delta G equals delta G0, which is positive. The reaction A to B cannot proceed in the direction as written. Rather, the reverse reaction can proceed. In other words, under a standard condition, fructose 6-phosphate can be converted to glucose 6-phosphate, not the other way around. However, when A concentration is much greater than B concentration, the direction of reaction changes. For example, when glucose 6-phosphate concentration is 10 times as much as fructose 6-phosphate concentration, we plug this number into the free energy equation, we get delta G. Now, delta G is negative. This reaction is spontaneous. These two conditions demonstrate change in reactants or product concentration can alter the direction of the reaction. If we give the reaction enough time, this reaction eventually will be in equilibrium. At equilibrium, delta G is 0. The free energy equation will look like this. If we move the delta G0 to the left side and define K as a ratio of concentration B over A, then we get equation for delta G0. Here, the R is constant. For reactions at physiological condition, temperature is also constant. The K can be determined by the concentration of glucose 6-phosphate and fructose 6-phosphate at equilibrium. We measure the concentration of these. Here are the data. Concentration of glucose 6-phosphate and fructose 6-phosphate at equilibrium. We plug this number in the equation. Then we get delta G0. This is how we determine delta G0 in the laboratory. Second conclusion, delta G of consecutive reactions are additive. For example, given a reaction A plus B to Y plus Z, it takes 2 steps through an intermediate C. Delta G for the first step is delta G1, and the delta G for the second step is delta G2. The delta G for the overall reaction is the sum of delta G1 and delta G2. As long as delta G is negative, the overall reaction can proceed as written. To elaborate on conclusion 2, here I'm going to give you another example. Given 2 reactions, A to B and C to D, the delta G for the first reaction is minus 3.0 kilocalorie per mole. The delta G for the second reaction is 3.0 kilocalorie per mole. How much is the delta G for the overall reaction, A plus D to B plus C? The answer is minus 6 kilocalorie per mole. In our body, ATP is the most common energy carrier. ATP can be hydrolyzed to release 1 or 2 phosphate, giving rise to ADP, and AMP respectively. When the first phosphate bond is broken, ATP releases 7.3 kilocalories per mole of energy. Similarly, ADP can also release a large amount of energy when the phosphate bond is broken. Here we have conclusion 3. Given a reaction that is not spontaneous, addition of high energy carrier such as ATP or GTP can make it spontaneous. This is why in our body there are so many reactions that require ATP or GTP. This is why ATP is important for life. Now we are ready to examine how ATP is made in our body. First, let's look at the mitochondrial membranes. Here is the mitochondrial outer membrane, and this is mitochondrial inner membrane. Here are proteins involved in electron transport chain and oxidative phosphorylation. These proteins are located on the inner membrane of mitochondria. When we zoom in, we will see 4 large complexes, 1 small protein, and 1 compound. The 4 large protein complexes are complex 1, 2, 3, and 4. 1 small protein is cytochrome C that shuttled between complex 3 and 4. 1 small compound is coenzyme Q that shuttled between complex 1, 2, and 3. Complex 1, also called NADH dehydrogenase. Complex 2, succinate dehydrogenase. Complex 3, cytochrome C reductase, or also called cytochrome BC1. Complex 4, cytochrome C oxidase, or sometimes called cytochrome AA3. Here, there's one complex very unique. This is complex 2. It sits on the one side of the mitochondrial membrane, facing the matrix, instead of across the entire membrane. This animation shows you complex 1, 3, and 4 work as proton pumps to pump proton from the mitochondrial matrix across the inner membrane into the intermembrane space, creating a proton gradient. Complex 1 oxidize NADH, reduce coenzyme Q, and pump proton to the intermembrane space. Complex 2 oxidize succinate, reduce coenzyme Q, but does not pump any proton. Coenzyme Q receives electron and pass it to complex 3. Complex 3 pass electron to cytochrome C by reducing the heme in it. Cytochrome C pass electron to complex 4. Cytochrome C is oxidized. Both complex 3 and 4 pump proton to the intermembrane space. Complex 1 to 4 contains prosthetic groups. Complex 1 contains flavin mononucleotide or FMN. Complex 2 containing flavin adenine dinucleotide or FAD. Both complex 1 and 2 also contains iron sulfur center. Complex 3 contains 3 hemes, 2 B types and 1 C type. Complex 3 also contains an iron sulfur center. Complex 4 contains 2 types of hemes and 2 copper centers. As shown in this picture, Heme groups is shown in blue and pink. The copper center are shown in brown. Complexes 1, 2, and 3 contain the iron-sulfur center. Iron form covalent bond with sulfur on cysteine residues from protein. NADH is derived from metabolic pathways such as glycolysis, TCA cycle, and beta oxidation. FADH2 is produced by succinate dehydrogenase and by the alpha-glycerol phosphate shuttle. Oxygen is delivered to tissues by hemoglobin. Here is integration with pharmacology. Electron and amytal inhibit NADH dehydrogenase, complex 1. Antimycin A is an antibiotic that inhibits complex 3. Cyanide, azide, carbon monoxide inhibit complex 4. Doxorubicin, an anti-cancer drug that we discussed before, inhibits coenzyme Q. To detoxify the cyanide, 3 drugs are effective. First drug is amyl nitrite. It acts as an oxidant to induce formation of methemohemoglobin, which sequesters cyanide. Second drug is thiosulfate. This drug reacts with cyanide to form thiocyanate. Third drug is hydroxycobalamin. It combines with cyanide to form a stable cyanocobalamin. Carbon monoxide binds complex 4 less tightly than cyanide, but it also binds to hemoglobin, displacing oxygen. Lack of oxygen and inhibition of electron transport chain leads to headache, nausea, tachycardia, and tachypnea, seen in carbon monoxide poisoning. As I described to you earlier, electron transport chains generate proton gradient. Proton gradient is relieved by pumping proton back by ATP synthase. Energy released in this process is coupled to ATP synthesis, and this is oxidative phosphorylation. Here is an integration with pathology. Leigh syndrome is a severe neurological disorder characterized by progressive loss of mental and movement abilities. It is caused by mutation in complex 1, 4, or 5. Here is integration with pharmacology. Oligomycin is an antibiotic. It inhibits ATP synthase by binding F0 region of the enzyme. Since electron transport chain and oxidative phosphorylation are coupled, inhibition of ATP synthase also blocks electron transport chain. Although electron transport chain and oxidative phosphorylation are coupled, some proteins and chemicals are able to uncouple these two processes. Therefore, they are called uncouplers. They include thermogenin, dinitrophenyl aspirin, thyroxine. They all act by destroying the proton gradient. Thermogenin, or uncoupling protein 1, UCP, is a proton channel. It increases the permeability of the inner mitochondrial membrane to proton, leading to a proton leak. This is an example of uncoupling protein. Newborns contain brown fat in neck and upper back. It acts as a biological heating pad. Brown fat is enriched with cytochrome-containing mitochondria. Upon stress hormone such as norepinephrine induce release of fatty acids from triacylglycerol in brown fat, which activates thermogenin. Thermogenesis creates proton leak, which generates heat instead of ATP. Synthetic uncouplers increase the permeability of inner mitochondrial membrane to protons. These include 2,4-dinitrophenol, a lipophilic proton carrier that readily diffuses through the mitochondrial membrane and carried proton with it. This creates a proton leak. Other synthetic uncouplers, such as aspirin in high dose, create a proton leak in a similar manner. When oxygen is partially reduced, unstable products called reactive oxygen species are formed. They react rapidly with lipids, proteins, and other substrates, leading to damage to lipids, proteins, and cell membrane. Reactive oxygen species include superoxide, hydrogen peroxide, and hydroxyl radical. Here is integration with immunology. Phagocytes, including macrophages and neutrophils, produce these reactive oxygen species to kill bacteria in the phagolysosome during the oxidative burst accompanying phagocytosis. Small quantities of reactive oxygen species are inevitable byproducts of the electron transport chain. These small quantities are normally destroyed by protective enzymes such as catalase. Here is the integration with physiology. The rate of reactive oxygen species production can increase dramatically under certain conditions. For example, during myocardial infarction, ATP levels will be low and NADH level high in a tissues deprived of oxygen. When oxygen is suddenly introduced, there's a burst of activity in the electron transport chain, generating a large quantity of reactive oxygen species, leading to reperfusion injury. Defenses against reactive oxygen species are particularly important in highly aerobic tissues. Tools include protective enzymes such as superoxide dismutase and catalase. In red blood cells, large amounts of superoxides are generated by the spontaneous dissociation of the oxygen from hemoglobin. The products are methylhemoglobin and superoxide. The processes that adequately detoxify the superoxide require a variety of enzymes and compounds, including superoxide dismutase, catalase, glutathione peroxidase, along with vitamin E and vitamin C. Low level of any of these detoxifying substances results in hemolysis. For example, inadequate production of NADPH in G6PD deficiency results in accumulation of destructive hydrogen peroxide. We will discuss this later. Hypoxia deprives the electron transport chain of sufficient oxygen, decreasing the rate of electron transport chain and ATP production. When ATP levels fall, glycolysis increases, and in the absence of oxygen, will produce lactate, leading to lactic acidosis. Here is integration with physiology. During myocardial infarction, myocytes swell as the membrane potential collapses and the cell gets leaky. Enzymes are released from the damaged tissue, and lactic acidosis contribute to protein precipitation and coagulation necrosis. Enzymes and proteins released from the damaged tissue can aid in diagnosis. They include creatine kinase, troponins, and lactate dehydrogenase. Mitochondrial DNA is composed of 37 genes found in a single circular chromosome. Among them, 13 genes encode proteins of the electron transport chain, 22 encodes tRNA, and 2 encodes rRNA. The 13 proteins involved in the electron transport chain are necessary for oxidative phosphorylation. Mutation in mitochondrial genes typically reduce ATP production, and hereditary mitochondrial disease have a global impact on the body. The tissues most sensitive to mitochondrial mutations are those highly aerobic tissues, such as nerves and cardiac muscle. Mutations that cause hereditary mitochondrial disease typically affect T-cell cycle, electron transport chain, and beta oxidation either directly or indirectly. For example, Leber's hereditary optic neuropathy is caused by defects in NADH dehydrogenase. This is directly linked to electron transport chain. Second example is mitochondrial myopathies. Here we will use Murphy syndrome as an example. Murphy syndrome stands for myoclonic epilepsy red fibers. It is caused by point mutation in the lysine tRNA gene. Mutation in this gene is indirectly linked to electron transport chain.